That's great of you. Right. Very good morning to the third session speaker, Angel Medina Vayo, and good afternoon to the participants. And we have with us a respectable registrar evaluation, Professor Mahadevan. Welcome you, sir, for this third session.
Thank you, sir.
I request Satish to hand over a bouquet. So we'll start our third session by introducing the third speaker session, Dr. Angel Medina Vayo, a reader in food mycology. Angel obtained his a PhD degree in microbiology in 2007 at the University of Valencia, Spain. He was awarded the prize for the best PhD thesis by the University of Valencia based on the novelty and quality of his research in 2008. Subsequently, he worked for almost 2 years in the food industry until he obtained a postdoctoral grant from the Spanish Ministry of Science and Innovation to work in the Applied Mycology group at Cranfield University, UK. He joined the university in January 2009. Angel has focused his research interest on the impact that environmental stress has on the functioning of fungi, mainly mycotoxin species. The mechanisms used for ecophysiological tolerance and the molecular basis of secondary metabolite production, especially mycotoxins, and other relevant metabolites for industrial applications. He has been developing research on ecophysiology, molecular ecology, and modeling of mycotoxin fungi growth and toxin production for more than 18 years. Angel has been involved in several projects to improve the storage and increase quality of small grain cereals and groundnuts. Dr. Angel has published more than 95 peer-reviewed journal papers, 12 book chapters, and his research credential, which says that he has an H factor around 26 in Scoopers and around 32 in Google Scholars. He combines his research with the role of director of the MSc taught program in agri-food. With this small introduction, I also would like to congratulate you. Recently, in September 2020, he has been awarded a British Mycological Society Berkeley Award for his contribution to mycology as early academician. Congratulations, Medina.
Yeah, thank you very much.
Over to you, Medina. Your stage is yours now.
Yeah, let me just show my screen. Well, first of all, thank you very much for the invitation to participate in this. And let me just know if you can see my presentation. Can you see my presentation?
Yes, we can see.
So let me just now put
Slideshow.
the view.
Yeah.
So can you see my presentation?
Yes, we can.
Or is this the preview vision? Oh, no, you can see that one, yeah.
Yeah, it is last
So
slide. Yeah.
Yeah, so let me just go to the beginning. Why it will be in the last slide? That's another.
To get a trailer of your talk.
Yeah, this is not the talk I wanted to share anyway. That's why it's in the last. That's why it's in the last one.
Okay.
Where is the? Yeah, this is what happens when you wake up at 6 in the morning, you know. This is what happens. Let me just close this. So can you see now anything?
No.
No, okay.
We
great.
can see you. Oh, yeah.
Well, that's unlucky for you. Let me just, because you know the computer just woke up in the morning and now it's trying to. Okay. I've got now. This is the problems of life
due to COVID.
Events. Yeah. So let's see now, share screen. We are, here it is. Hallelujah. I found it.
Spanish.
Yeah. What about now?
Yes. Yep.
Here we go. Fantastic. Okay, sorry for the technical problems. Yeah, it's again, I wanted to reiterate my thanks to Senevisa for the invite and to the whole university for giving me this opportunity. And, you know, for me, it's always exciting, and it's one of the things that I love the most, explain the people what we have been doing here at Cranfield, and, you know, explain the research that we are doing with regard to mycotoxins and climate change, because this is an area where I'm very passionate for the last 8 to 10 years. So the title of my talk today is going to be Climate Change and Mycotoxins, and we're going to be reviewing slightly the implications for food safety. I could talk about this for hours and hours, so I had to decide, you know, which part I wanted to show. I hope it's interesting for you. So what am I going to be talking about? So I'm going to be talking, first of all, I will, just in case someone in the room doesn't know what mycotoxins are, I'll give a quick reminder. Then what do I mean when I say climate change and as a changing world? Then what are the most important environmental fluctuations that are important for crops and toxins? Then I'll review what do we know so far, and I will go then to the research that we've been doing, and then we will look at the future. And, you know, I'll leave you with a big question for you to think about. So what are mycotoxins? This is the most important thing because it's in the title of my talk. So mycotoxins are naturally occurring toxic secondary metabolites that are produced by fungi when they are growing in the field, or they are growing in while we are storing grains. What is the main problem of mycotoxins is that the molecules, the chemical structures of mycotoxins are highly heat stable. So they are very difficult to destroy. So once we have those mycotoxins in our seeds or in our ingredients, or in any of the ingredients that we are going to use to produce our food, then it's going to be difficult to destroy. Then, which fungi produces mycotoxins? So that is another big problem because it's our genera that are very common, like Aspergillus, Penicillium, Fusarium, and Alternaria. So wherever you are in the world, you can find this type of fungi around you. And then something particular about mycotoxins. So the mycotoxins can appear or can occur in the field or in storage. So they can be there throughout the whole food chain. And then something very important that some people sometimes forget is that the absence of mold, so although your food at the moment when you are going to eat it is not moldy and you cannot see that is moldy, the mycotoxins are already there. And they are dangerous because you can't see them, you can't smell them, and they don't have any taste. So you never know whether the food that you are eating has mycotoxins or not. So what do we mean by food security? So food security is the ability that we have to feed all the population in the world. And this thing has been suffering some impacts in the last 5-10 years because the price of staple food all around the world is increasing. So that's putting a lot of pressure in food production. Then that is impacted by climate change. So that is putting a lot of pressure in food supply, in the quality of food that we can eat. And in the sustainability of the food chains around the world, I think we have now a fantastic example with COVID of how weak our food supply chains are. Because here in Europe, for example, that you will think that everything will work fine. We had few days in which we couldn't find any vegetables in our supermarket. And then, you know, we know that all these climate change things, all these changes in with droughts and rainfalls, etc., they are putting pressure in the way that we produce food, and they impact on the stress of the plants and the fungi that is growing on the plants and is producing diseases. And it's very important to know that already 25% of the production of cereals in the world is spoiled because of fungi. So with all these other things happening at the same time, we expect that we are going to have problems in the future. So what is that we call climate change? Climate change is already in the news. When we are looking at the news here in the UK, for example, we can see, you know, fires in Greece, we can see drought in the UK. So the normal thing when you think about the UK is that it's always raining, but so then how is possible that we have droughts if it's always raining? It's not raining that much now at the moment as it used to rain. Then we've seen in the news as well, floods in Japan. We've seen cases of extreme temperatures in some areas of the world. We've seen the reduction of oxygen in seas. We've seen in New Zealand, you know, floods that have been extraordinarily big and that are destroying everything. We all know that it's been said that in Greenland, you know, the glaciers are melting very fast. And, you know, all over the world, there is destruction and natural disasters. And when we see each of these disasters, this is impacting some of the food that we can eat. And one of the last ones that was really, really big because of an extreme drought and very high temperatures in Australia in 2019, they had bushfires, and there was an increase in temperature, and you know, it was a tremendous episode of like giving examples of this climate change. So when we say climate change now, and what does science says about climate change? So one of the main factors when it comes to explain climate change is the increase of CO2 in the atmosphere and the increase in temperature. So temperature and CO2 will increase. So in this graph here, I'm showing what is the level of CO2 that is currently in the atmosphere. So while in the 80s, it was around 340, at the moment, I checked yesterday in NASA and NOAA, and at the moment it's 416 when it used to be 340. And then when it comes to projections, it doesn't look good because it shows that the temperature in our planet is also going to increase. In some projections up to 4 degrees, and in other projections, in the best projections, even by 1 or 1.5 degrees, which is not good because it kind of impacts our food production. Something that is gonna also change is the episodes of drought and the episodes of floods. This is gonna be more radical and it's gonna be local. So in some places it will rain a lot, or in other places where it used to rain, we won't have that amount of rain that we used to have, again, affecting the way that we produce food. And then this, as I said, is impacting food security, and in a recent report in 2018 by FAO, some areas of the world were impacted as, you know, with scored with high threat of food security, and India is included in the high value. So I thought it was very important to show today. So what is the resilience of food crops? So when all these things happen, what is going to happen really to those foods and the foods we produce. So in rice, for example, and rice is a very good example because it's eaten all over the world and in high amounts. The rice needs 12 to 13 degrees for developing the canopy. Doubling the CO2 will improve the photosynthetic rate and also increase rice biomass by 20–25%. So if we stop there, that'll be good news. However, if we combine elevated temperatures and CO2, then this makes the yield reduce 15%, which is no good. In the case of maize, another staple around the world, mainly in Africa, so there will be an increasing need of irrigation. Africa will have less rain. So then if there is no extra irrigation, there will be a penalty of 15%. And in the case of maize, there is no problem with CO2. However, now in wheat, also very important in some areas of India, and when we increase CO2 and we increase by 4 degrees the temperature, the average temperature, the outcome is around 20% of penalty in yield. So as you can see, these are big figures that could impact the way we eat. So now let's go a little bit more to the microbiology. So now that I have set the scene and we know what are we expecting with climate change, let me just show you, and let's change to the microbiology. That is what you all are interested. So our focus in my group is mycotoxins. As I told you, mycotoxins are very important because they can produce diseases in animals and in humans, mainly cancer. And as I told you, you cannot see them. So we have been doing research in ecophysiology of mycotoxigenic fungi. And for that, what we've done is we have considered how different interacting environmental factors affect the growth of fungi and also the production of mycotoxins when they are either in storage or they are in the field. And how we have approached this problem? We have approached this problem doing something that we like to call molecular ecology. So what does molecular ecology mean? So we know that the environment in which the fungi are growing has a high influence in the way the genes inside the fungi are switched on or switched off. So it's been already published and is known that parameters like water activity, the pH, temperature, the nutritional substrate, and CO2 are able to control those metabolic gene clusters. And why is this? So normally inside the fungus, when it comes to the production of the mycotoxins, all the genes that are needed for producing a toxin are normally clustered together, and they depend on the same regulatory genes and catabolite repression. So what we've done is check how these environmental factors affect these pathways so we can understand how climate change and how all these changes will change the way in which the fungus is going to respond to climate change. So another thing that I want you to have in mind is that if you've been reading something about plant pathology, you will have heard about the triangle for disease. So to have a disease, you need to have the environment, the right the conditions, you need to have the crop, and you need to have the microbes that are going to produce that disease. So normally it was described as a perfect triangle. You need the collaboration of the three to have the disease. However, with climate change, a lot of pressure is going to be put in this triangle. So the triangle is not going to be a triangle anymore, because there are going to be, environmental pressures. There are going to be pressures on the crop, and there are going to be pressures on the microorganisms that will change the way in which diseases are developed now. So if you combine everything together, and we look at now interaction of environmental conditions with our fungi, so a lot of interest has been, and a lot of publications have been published in the last 10 years. And most of those publications have taken into account two main factors. And we're going to do a little review on that. So it's one of the main ones is water activity. So water activity is the available water that microorganisms will have to grow in the grain or in the plants, and then temperature. So what are the effects of these two parameters? So if I put a big example on wheat, for example, so there's been a lot of publications in the EFSA and in Nature as well. And what they have shown is that with increases in temperature, what is going to happen is that the harvest date for wheat will change. And what that means is that the harvest will be earlier than it is now. But that, on the contrary, has an effect, and is that the amount of fungi that will be able to grow and the amount of fungi that will be able to contaminate wheat in very important stages when they can produce disease will also be impacted by increasing. And that will mean that there will be a chance to have higher toxin contamination. And when we look, for example, in maize, another example, to research from our colleague Paola Battilani, also published in Science, in Nature Scientific Report, sorry, what they did was in 2016, they looked at increases in, they looked in Europe, and what they did is they measured the probability of having aflatoxin B1 in maize in Europe in the current scenario, and then with the current temperatures, you have the map that you are watching on the left of your screen. So as you can see, little blue and little red dots, basically in Spain and Italy and in the Greece area. Then they modeled with predictions that they had available until 2000 and until the year 2079. They model what will happen if we have an increase in 2 degrees, and then you have the map that you have in the center. And as you can see, when you compare the first map with the second map, you can see that the red is increasing at the moment, and you can see that many areas of Spain, Italy, and Greece will have many more, much more probability of having toxins. And then if we go to the 5 degrees scenario, it's not only that we have the same countries. When we have a 5-degree scenario, we can see that the problem with aflatoxin could even expand to the north of Europe. And although maybe we don't have so much red in the +5 scenario, what we have is a much, much more extensive area of crops that could be contaminated with these fungus. So indeed, very important, and that could pose very, very high problems because aflatoxin is one of the main and most toxic mycotoxins around the globe. So this is basically kind of a brief view on what has been published so far and what has been our contribution to this. So our contribution from Cranfield has been to study the 3 main factors at the same time. So as I told you before, that main research was done with water and temperature. However, at Cranfield, we've measured we've mixed water, temperature, but also we have included in the picture CO2, because CO2, as I said at the beginning, is an important gas that is going to increase by doubling or tripling by the year 2100. So I'm going to use as a case study for my talk today the work that we've been doing with maize and Aspergillus flavus. Why is that? Because it's a fantastic example of what's happening around the world. So in normal conditions, when we have the weather as it used to be 10 years ago, the main fungus that will contaminate maize plants will be Fusarium verticilioides. So Fusarium verticilioides is a fungus that produces mycotoxins, but the mycotoxin that produces is not very toxic, let's say. You need high amounts of fumonicins for it to be toxic. However, when you have an increase in temperature and drought conditions, the maize plants are very, very sensitive to drought. And what happens is that immediately the plants, instead of being contaminated with Fusarium verticilioides, when you have drought, they become contaminated with Aspergillus flavus. And then Aspergillus flavus is much more complicated. First of all, because it's much more resistant, but second, because it produces Aflatoxin B1 and other aflatoxins. But aflatoxin, just for you to have an idea, is the most potent carcinogenic molecule of biological origin known. So there is nothing more toxic than aflatoxin that is from biological origin. It has been related very clearly with hepatic cancer. And if you eat enough of it in one go, you will have fulminant cirrhosis. Then aflatoxin B1 among the whole family is the most toxic. And the International Agency for Research on Cancer has put this molecule on the top group as definitely carcinogenic for humans in group 1A. So because of this, there is very strict European legislation for these maximum levels. And I'm proud to say now that more than 150 countries around the world have now legislation for these toxins. Where we can find them? We can find them in maize, in cereals, in dry fruits. Now there are a lot of problems with figs, and also a lot of problems with nuts, like pistachios, peanuts, and other type of nuts that can become highly contaminated. So when we did the first experiment in maize, you have here in this histogram, you have here the amount of toxin in the y-axis, and then you have different conditions. So what we did is we took maize and we put maize under different environmental conditions. So we have here 30 degrees, at the, then water activity 0.91, so very dry maize. And then the amount of CO2 that we have now, the one that we expect in 2030, and the one that we expect in year 2100. So 350, 650, or 1000. So what you can see is that in the middle here, I'm just pointing out to the main area of the slide now. So when we have 30 degrees, but slightly wet, which is water activity 0.99 maize, what we see is that this is the amount of toxin that we have now, which will be lower than 1000 nanograms per gram of maize. But when we use the conditions where we have mimicked a change in weather, mimicking climate change conditions, the amount of toxin that is produced increases almost 600 times. So this is really, really bad. And it happened not only at 30 degrees, but also at 37 degrees, which is when we are mimicking not only increases in CO2, but also the increases in temperature. So in many places of the world where maize is grown, there are peaks at 37 degrees during the day. And what we have demonstrated here is that the fungus with CO2, and if we have together CO2 plus temperature, it will go from producing almost no toxin to produce high amounts of toxin. This is not good news. What we did is, as I said, we use a molecular approach. So we looked at which genes within the biological pathway of aflatoxin will be related to this production. And in this table that you have here, we found that when looking at the different genes, we found a quite good correlation between the gene AFLR and the amount of toxin that was produced. So you can see here that when there was an increase in AFLR transcription, that resulted in increase in aflatoxin in most of the cases. And AFLR, if you remember a few slides ago, is one of those regulatory genes of the whole pathway that I explained before. So it's clear that when we have CO2 and increases in temperature, so when CO2 is in the picture, FLR, something happens with FLR that is telling us and is making some changes in the way that the fungi produces mycotoxin. So when we did this table back in 2017, we were thinking, wow, this is, there is much more here that we could not understand at the moment. So I went, to a conference or, giving one of these talks, like the one that I'm giving to you now, showing these slides and everything. And then I met a nice guy from the United States Department of Agriculture. And, I was showing them this data and they told me, wow, I mean, why don't we collaborate together? And, we can do RNA-seq. So instead of just looking at one gene or the 2-3 genes that we were able to look using PCR, they offered us, oh, why don't we look to the whole genes, all the genes that are there in the fungi to produce the toxin. And then I thought, wow, this is nice. Why not? And in the same conference later, I spoke with a guy from Vienna, and instead of doing only the analysis of aflatoxin, he offered me the possibility to do analysis of all metabolites that were produced by the fungus when it was growing on maize. So then I put together this little cluster of people collaborating together. And what we did is we did RNA-seq that was done in States, and we did metabolomics that was done in Voku, and then we did all the environmental work at Cranfield. So I'm gonna show you some of the results that we had. So now here you have a heat map. So instead of having only one of the genes like AFLR, that you can see here, AFLR or AFLS, which will be the regulatory genes, we have the list of all the genes that have been involved in the production of the toxin. And if you see here, you have the temperature, the water activity, like mimicking drought, and the CO2 concentration. And here you have 350, 650, and 1000. So we use exactly the same type of experiment, and then we looked at how this was going up and down. And as you can see here, there are two differences, and I want to point out two lines. This is at slightly moist maize. This is the current conditions, so high gene expression in some of the genes. And this is what happens when we increase the amount of CO2. So definitely a big change affecting, but as you can see, AFLR is one of the ones that is affected, and the other ones look like they are less less transcribed. So then we went a step further, because with our colleagues in USA, we know that aflatoxins is a secondary metabolite cluster, but in Aspergillus flavus, there are other pathways that have been described as secondary metabolites. And there are 55 of those potential metabolites. Some of them, we know what they produce. In some of them is not known. So what we did in this slide is using RNA-seq, we looked at the genes inside these secondary metabolite pathways. So the secondary metabolite pathways are all metabolites that the fungi is trying to produce in order to survive or to produce something that will help the fungi. And we look at the effect of temperature, we could see that, like, for example, an effect on aflatoxins and an effect on other mycotoxin, which is cyclopiasonic acid. When we looked at the effect of water activity combined with CO2, we also looked at with CO2, there was an increase in production of cyclopiasonic acid. But then we were able, using a new methodology, to study the interaction between the three factors at the same time, which means when I have CO2 in the picture, does it modify the way that the fungus reacts to water activity? Or does it modify the way that the fungus reacts to temperature? And the answer, as you can see here, is for aflatoxins, yes. And you have here a positive response, 2.9. But what we observed was something even more worrying. And is that for another mycotoxin that is less toxic than aflatoxins, but is also quite toxic, there was a very, very, very big number here, 8.1. So that told us that we should look also to this other toxin, because under climate change conditions, there was a big chance that this toxin could have important changes. So that's what we did with collaboration from the other partners. So instead of looking only to one molecule, aflatoxin, we looked at the whole set of toxins, and this is the results here. So when we have current conditions will be these ones here that I'm pointing out, 30 or 37 degrees, 400 ppms of CO2. And you can see that the blue line here is very long. The blue line here is very long. We only have a little bit of red and a little bit of yellow. However, when we have increase of CO2, as it was said by the gene expression data, can you see here how big the yellow is? So what is happening is that, as happens in the genes, so we've been able to corroborate what we were explaining, what I was explaining before. So by looking at the genome and looking the interaction of these three factors, we can see how CPA, cyclopiasonic acid, is going to increase in production. It won't happen, as we can see here at the bottom, when we have maize that is dry. But if we have a maize silo where, or a storage conditions where slightly amounts of rain could go in, or we have, for example, some pests that are producing, and when reproducing, producing water, and they increase the amount of water that will be available, then we might say now, oh, there is no problem. The amount of aflatoxin B1 has been reduced. However, the amount of cyclopiasonic acid will be increased a lot. So this is another toxin that we will maybe need to look at the future. So as you can see, big changes depending on the climate change. So in conclusion for this first part of my talk, so I think I want you and I want to remember that CO2 is affecting the production of aflatoxin. So CO2 is affecting the molecular response that, that Aspergillus flavus has, and under the stress conditions and water activity, under temperature stress and waterability stress, CO2 modifies the way that the fungus responds. Then the interaction between the three, we have shown that affects the metabolism of the fungus. You've seen how it has changed the secondary metabolites that it's producing. And then the interaction of the three factors might lead to changes in other toxins, not only in aflatoxin, but other toxins. So now the question that some of you might have is, okay, you know, this guy, this Angel that we just met might be obsessed with maize and aspergillus flavus, and he's, you know, putting the data here so it looks like it's going to be very bad. But what will happen with other fungi or other grains? So I've had the liberty to put 2 or 3 more slides for you guys, because when we looked at another fungus that produces another very important toxin, that is ochratoxin A, and we did a similar experiment, increasing CO2 here. So blue lines, as you can see, keep going higher and higher and higher than the orange. So for this fungus, so other type of grain, so in this case more wheat than maize, it will also happen that we'll have more toxin when we change the conditions. Another one is Aspergillus carbonarius, very important for production of raisins, sultanas, and grapes and wine. And what you can see is that in some cases, the orange is higher than the yellow. In here, for example, and here, very clear changes. Another big disease now in Europe is for oats. Oats are contaminated with Fusarium lancetiae, and the big problem of this disease is that it doesn't have any symptoms. So you cannot differentiate between contaminated and non-contaminated oats. And when we did this in a big project that we have, look at this, we have increases in 5 times of one of the toxins, 4 times other of the toxins, but it's even worse here when we have from nothing, so no production, to increases on 264 times. So very, very big, a very, very big problem, not only in the example that I just gave to you, but also in other areas and for other toxins. So don't think that I've just picked up an example to make you worry. This happens in a huge diversity of different scenarios. So now let's recap. And you know, what I wanted to do at the end is to make you think a little bit, and you know, and let's comment together this. So what we are looking, and with the results that I've shown to you, it's clear that there will be an increase in plant diseases and pest reproduction, and the interaction with plants may increase mycotoxin contamination. I think that we have answered yes, and I've tried to give you some evidence for that. Then there will be significant potential impacts on the nutritional value of food and feed crops. As I told you, with maize and with rice, for example, there will be changes, and that could lead also to mycotoxin contamination. That is clear. However, there are many things that are not that clear. So, for example, we have been one of the first ones on using metabolomics, and I have showed you the data that says that there might be changes in mycotoxins produced by the species, and that the ratios might change. So only few papers and few studies have been looking at that, so more data is needed. So more data is also required on free versus bound mycotoxins, MAX. This is a big area now, and at the moment there is no data about it. Nobody has studied it. Then the use of sustainable mycotoxin control systems. I haven't spoken about this today, but it's clear that maybe with the changes in weather, this might mean that the current systems don't work anymore. So more research will be needed. So maybe the fungi that we are studying now is not exactly the same as we're going to have in 30 years, because they will be surviving and surviving under these conditions for many years. So there might be an acclimatization of those fungi, and that is a probable thing because we've been running some experiments now, and I can tell you that acclimatization has a big impact and is no good. It looks like when you acclimatize, you have even more toxin than in the experiments that we did at the beginning. So that puts us in a very bad situation, because as you know, to change a legislation in food takes years and few years, and doesn't wait for the legislation to be ready. It looks like they are already at this time producing more toxin than we used to have in the past. So I'm sure that legislation is not keeping up fast enough. And our control strategies and the way that we control now mycotoxins definitely will need to change to take into account these things that I'm telling you, and climate change, because of the effects of climate change. So I have a question for you after I've been speaking now for 41 minutes. So do you think, after what I told you, we know enough about mycotoxins and climate change? So I hope everybody has arrived to the same conclusion as I did, and I don't think so. We need much more information. We need much more research. I hope from the students that I have here today, some of you will be interested in this exciting area of mycotoxins and control. I know that SREP is working on that in Mysore and looking at biocontrol with bacteria. Fantastic. This is how I got to meet him in the first place. So many more things need to be done in this area. And then obviously, this is not a work of a man in a lab. So this has been a fantastic collaboration in the last 10 years with many people around the world, with money from many different sources. And I would like to thank all my collaborators, mainly in the Applied Mycology group, Professor Nares Megan, that has worked a lot in this, and all our postdocs and the people in North Carolina State University, the USDA, in Boku that did the metabolomics. I'm very thankful to all of them. And if you want to know more, follow us in Twitter. We post about all the things that we are doing. And thank you very much for listening. And I'll be very, very happy to take any questions that you might have.
It was fantastic and informative talk. Really igniting. You explained it in a proper way, from basic to the burning issue of climate change.
Yeah.
So you have already asked some questions during your talk at the end, and you have also answered also. However,
okay, great.
There are some questions which come from the chat from the participants. Like one says, is there any possibility to suppress the mycotoxinic gene clusters due to the climate change? Like, as you said, it is going to express.
Yes. So what is happening, is that fortunately, not all Aspergillus flavus that you can isolate from the environment will be able to produce the toxin. So some strains of these fungus don't have the genes. So once they have the genes, it's very little that you can do unless you use very hard things or whatever. But what we can do is, and what is done around the world with this pathogen, is biocontrol. And for that, what they are using at the moment is to spread in the field some other strain of Aspergillus flavus that won't have the genes. So then what happens is that because you increase a lot the population of fungus that doesn't have the gene cluster, then you'll have less of the one with the gene cluster that could be able to arrive to the crops. Then if you ask me about suppressing the gene cluster, there are some research now on natural compounds, plants, lactic acid bacteria, of many types of compounds that are trying to control the amount of these toxins that are produced. And some of these compounds will, in general, reduce the growth of the fungi, but others will maybe affect the way in which the metabolism of the fungus works. So they could be possible. However, the most important thing, as I said, is that we need to make sure that the systems, as we are developing them now with the current environmental conditions, is the same system, is the same molecule. Is that molecule going to work when we have 2 or 3 degrees in temperature extra? Does it work when the conditions are not the same and when we have more drought conditions? Because what we've seen is that some don't. And when we are developing these new systems to control toxin production, we need to be aware of these environmental changes, looking what is going to happen in the next 10 years. So when we develop a product or a system, we need to make sure that it will work in the long run. Otherwise, why to spend the money on developing something that will work for 3-4 years and then it won't work anymore? That's my point.
Yeah, you are right. You are right. And one more question. So how would you overcome the mycotoxinic fungi or mycotoxins when they enter into the human systems? So if they're outside, okay, but how to control them once they are into the systems?
So into the systems, you mean inside the body?
Yes, sir.
So normally these fungus are generally plant pathogens. So although we might be breathing them and having them when they arrive to our lungs, if we are healthy, they should be fine. We should kill them and we won't have any problem. However, Aspergillus flavus has been described in some places as a nosocomial disease, which means that if you've had an operation or you are immunocompromised after being in hospital or whatever, and you get infected with Aspergillus flavus, it could produce mycotoxicosis as well, and it could produce infection. But the main thing of these fungi is that they cause disease, but mainly in plants. And what affects us is not exactly the fungus, is the mycotoxin that they release on the food products. That's very important to remember.
Yes, I'll take the last question from the participants. So what might be the possible factors favoring mold growth in canned foods and dehydrated foods? There are some reports which says that fungal growth in canned foods and dehydrated foods.
Yeah, so normally in the one of the things that make Aspergillus flavus and this type of fungi very worrying is that they have an incredible ability to survive. And fungi, the reason why I started working with fungi is because, fungi is really amazing when it comes to survive in dry conditions. So they can survive in much drier conditions than almost any other microorganism. And that makes them the thing or the organism that will spoil most of that dry food, although it's prepared, or then also it makes them produce spores that can survive in cans or can survive in food that is expected to be preserved for a long time. And if the process is not completely right and the temperatures used are not completely right, they will be able to germinate and grow even after years and years of being dormant. So you have to be very careful. And, in dry fruit, in dried fruit, food or feed, the most important thing to control, if you want to have your food or feed stored for a very long time, is that the water activity of your food needs to be below 0.7. So that is, for example, in wheat, is around 14% of relative humidity or moisture content, 14% of moisture content. In wheat, for example, again, is around 12, 14%. So when you store all these ingredients, you need to make sure that the moisture content at which you are storing them has to be right. So the water activity is below 0.7, so you can be pretty sure that no fungus is are going to grow there. Or no fungus is going to grow. But if you have pests or you have any water ingress in your silo, then the whole ecosystem might change, and with just few drops, then you can start having fungal growth. So you have to be very careful with that.
Yes, thank you. And there are a few more questions which can be answered in your Twitter or through email. So probably we will ask them to contact you through email.
Yeah, that's fine. You can contact me whenever you need, and I can send you the answers or record a short video and send you the answers. So no problem at all.
Okay. So now I'll request our chairperson, Professor Lakshmi Devi, to propose a vote of thanks. Over to Madam.
Hello, Dr. Angel Medina Vayo. I thank you very much for delivering a nice talk, an informative talk on climate change and mycotoxins. Okay, and your talk, apart from that, your discussion and answer for our queries was very much relevant and very informative. I thank you once and again.
Thank you very much.
Thank you.
Thank you, sir.
Thank you. Thank you, guys.
Yes, thank you.
Thanks. Bye-bye.
Bye. ಈಗ ಅದೇ ಇವಾಗ ಹೇಳ್ತಿದ್ದೇನೆ. Hello, good afternoon all of you. Today we heard 3 talks from eminent international speakers from 3 different countries on different aspects of microbiology. First, the lecture by Dr. Ram Savan, who spoke on heterogeneity of immune response against viral infections, was very informative, and the University of Mysore is thankful for the speaker for accepting our invitation and delivering the talk in the webinar. And the second talk was on food safety in hospitality industry, reality, consumer expectations, delivered by Dr. Gokul Shankar. So we, the Faculty of Microbiology Department and the University of Mysore, wholeheartedly thank you for your participation. And more than that, your presentation was very nice, not only informative, and also you have explained the role of food microbiologists and also the role of food safety officers in forming a policy apart from health, hygiene, all these things. You have focused more on the present day scenario in the food industry, sir. And the third lecture is on the Angel Medina Vayo delivered the talk on climate change and mycotoxins. I thank the speaker for accepting our invitation and delivering the talk in the webinar series. And also, he accepted to deliver the lecture at the very early in the morning, that is at 7 o'clock in India. In UK. And he was very enthusiastic at the early hours of the working hours in UK. And I thank the speaker wholeheartedly for his active participation in discussion by giving solution to our questions from the audience. And I thank our Honorable Vice Chancellor, Professor Hemant Kumar sir, our Registrar, Shivappa sir, and also our Registrar Evaluation, Dr. Mahadevan sir. And I also thank our department senior professors, Professor Shubha Gopal, Professor Satish, and Professor M.Y. Srinivas. Because of all their ideas, the present day webinar series is very much successful. I thank all of you for your efforts. Thank you very much, dear esteemed colleagues. I thank the Professor Chandranayak, the coordinator of the Vignan Bhavan, for all his participation and support for the international webinar. Thank you, Professor Chandranayak. Finally, I wish to thank all the participants for attending this webinar and also actively participating in discussion part and asking questions and getting answer from the three distinguished, eminent international speakers. I again thank one and all. Thank you very much.
Thank you, ma'am.
Thank you.
Thank you, one and all.